Hello, everyone. Thank you for joining us for today's webinar, Expediting Biologics Drug Development, Downstream Strategies to Accelerate Preclinical Development Timelines. I'm Leslie Bebko, your host from Bioprocess Online and Life Science Connect. With me today, I have Sunyoung Kim, Principal Scientist and CDO Downstream from Samsung Biologics. Over the next 45 minutes, he'll be discussing the utilization of historical data in the downstream process to expedite rapid process development and the implementation of effective strategies to shorten the timeline in technology transfer processes, and will analyze case studies demonstrating the successful application of bispecific antibodies as demonstrated in real-world scenarios. After his presentation, we'll be holding a Q&A session. Feel free to submit your questions as we go along. We'll get to as many as possible, and any that we can't get to, we'll follow up on after the event. If you run into any issues, please try refreshing your browser first, and if you need additional support, submit a question via the Q&A. Finally, this webinar will be available for on-demand viewing, and you'll receive a link via email as soon as that's available. Now, with all of that said, I'm going to turn it over to Sunyoung to get us started.
Hello, I'm Sunyoung Kim from DSP Group in Samsung Biologics, serving as Principal Scientist. Today, I'm excited to be a part of this webinar and present on Expediting Biologics Drug Development. I will focus on downstream strategies to accelerate the preclinical development timeline. I will firstly share some light on Samsung Biologics' background. Next, I will demonstrate to you what our downstream process group has been working on to accelerate the timeline with several case studies. Introducing our background and history, Samsung Biologics has always stressed that the customer's needs should be our central focus, and in meeting their needs, we have pushed our effort into raising our capacities to accommodate top-tier quality productions. Ever since Samsung Biologics was founded, it only took 5 years to establish 180 kiloliter of cGMP capacity. Our growth rate has only been rising exponentially since then. Samsung Biologics also has over 110 clients with a nearly perfect 98% batch success rate. We have accumulated over 260 approvals from various world agencies. The services that we provide cover everything from the early developmental stage, as well as the DSDP manufacturing and biotesting services. Samsung Biologics began CDO services in 2018 and has shown remarkable growth, providing our services to over 90 clients with proven quality. At the beginning, we solely focused on cell line, process, formulation, and method development. But then we realized that many of our clients would value supporting services in late-stage discovery. So we have been expanding our scope of the service with tech platforms, such as Developing, which is an in-house candidate scoring platform, and SDUAL, a bispecific antibody platform in 2022. We also launched S-Choice, a transient expression platform using S-Choice, our very own proprietary cell line, and S-Clean, a platform for glycosylation modulation. So before diving into our strategies to accelerate downstream processes, let's first address the challenges we encounter specifically within the downstream process of biologics development. With the demand for shorter development timelines, drug developers are increasingly pressuring contract development and manufacturing organizations to complete processes within a mere 3 months without compromising quality. Additionally, as molecular structures become more complex, traditional molecular antibodies are giving way to intricate forms such as fusions by specific antibodies and antibody fragments. Furthermore, in order to reduce production cost and scale efficiently, higher yields are necessary for investigational new drug submissions. These challenges highlight the collaborative effort required between drug developers and CDMOs to overcome these obstacles. To address these challenges, the DSP group in Samsung Biologics is implementing a strategy that integrates our historical experience with a meticulous approach for resin selection. This targets accelerating the development timeline while ensuring high-performance results. This approach is straightforward, but highly effective in optimizing both quality and timeline. Without our historical data, selecting the right resin for the project would be ambiguous and time-consuming. However, by leveraging our understanding of molecule characteristics through in silico prediction and chemical testing, we can narrow down the selection of resin candidates, shortening the timeline significantly. Here, I would like to give you a glimpse into how we are leveraging accumulated historical data. Upon analyzing results from various projects, we have observed net charge differences in impurities versus target protein, which we can incorporate into downstream process development. On the right side of the table, we confirm the charge value of partial molecule under different conditions, such as varying pH levels. Also, for resin selection trends, we observed that using IX resin was optimal for project A2H, while hydrophobic interaction chromatography or mixed mode chromatography has preferable for project K2Q. With such an insight, when we encounter new molecules, we can report to similar experience from previous projects and rapidly identify successful method development approaches. We can also get the information for determining the most suitable type of resins through conducting required physical and chemical tests. We have collected elution patterns from our previous projects using a standard HPLC method. And we have known that this elution pattern analysis gives us insight to select the right resin when we compare that of new molecules. With the solubility test, by monitoring the level of aggregation throughout different levels of hydrophobicity, we can draw on the hydrophobicity of the molecule. In the case of Project A, because the concentration declines toward the higher level of hydrophobicity, we can find that the molecule has relatively greater sensitivity for hydrophobicity, prompting consideration for the weak hydrophobic condition in chromatography. Therefore, we can get an insight to select the right resin and operational chromatographic conditions using our historical data. Now, let's examine the performance result derived from our case studies. The first case presented here demonstrates the successful completion of resin screening through the application of our strategy. This project is for FC fusion protein, where we compare two scenarios, one applying our strategy and the other without. As illustrated on the left side, we tailored the process workflow and conducted the first column stage using hydrophobic interaction chromatography. Our findings revealed that the applied strategy resulted in optimal performance, achieving around 42% yield with enhanced purity levels. In contrast, the non-applied case showed a very low yield. The next case that I'd like to explore here emphasizes the importance of not only selecting the right resin, but also optimizing the condition for resin performance. Drawing from our historical knowledge, we identified the cation exchange resin candidate based on charge values. Additionally, we determined the right pH level for optimal performance, which is possible because various pH conditions are included in our resin screening. In this instance, pH 6.5 is shown to be the optimal performance, resulting in substantially higher yield and purity. The final case discussed here involves a bispecific antibody developed using our proprietary platform, SDR. Our primary goal for this project was to maximize purity levels, ultimately achieving 99% purity within our platform. Our strategy for resin selection significantly contributed to this achievement. So in summary, our DSP group applies this approach in the early stage, drawing on historical knowledge and tests to understand molecular characteristics. Typically, we recommend to design the downstream process, including one or two resins from resin screening. The downstream process will be finalized by defining the optimal operational range of each resin through DOE study. As our DSP runs the development through a rigorous procedure, our past experience of running projects have ultimately led to either the retaining of or increasing both yield and purity in comparison to the feasibility stage. So to wrap up, we believe that each stage and aspect we implement and the way we implement will constitute the strategy best fit for the client. The downstream process flow can be set early stage based on our understanding of molecule characteristics through in silico analysis combined with historical data. By thoroughly understanding the accurate picture which these tools paint for us, we can find the most optimal resin quickly high yield, reducing time taken. Samsung Biologics has accumulated the extensive experience and data to support all these features and demonstrate to be capable of developing a robust and reproducible process for different type of molecule. With the technical strategy we are implementing, our team at Samsung Biologics is dedicated to accelerating our timeline while maintaining high performance. Here are the key factors contributing to this endeavor. First, effective communication is one of the key aspects of our service. Our team fosters open communication across all functions based on scientific and data-driven insight. Next, we leverage high-throughput systems, which are designed for speed and cost effectiveness. Additionally, we have our robust proprietary platforms. We have developed a range of platforms and we have continuously innovating with new ones. Those platforms enhanced our process method development capabilities. Another crucial aspect is our focus on manufacturability evaluation. We prioritize assessing manufacturability early and enable us to identify and address potential issues before they become major obstacles. This proactive approach streamlines tech transfer and minimizes risk, ensuring a smoother development journey. Last but not least, raw material management is key. As we take proactive measures to managing raw material throughout the development and production cycle. This proactive approach helps us eliminate a supply risk and maintain a steady workflow. So thank you for joining our webinar today. This was Song-Young Kim from Downstream Process Development at Samsung Biologics. If you wish to explore more about our services and or discuss about your developmental journey, please participate in our survey and tell us your needs. Thank you.
Song-Young, great presentation. Thank you so much. If you have not had a chance to submit your questions yet, please do so now. We'll get to as many as we can. And as he said, if you would like to fill out the survey and be contacted, please do so. And also, we do have some content available, some related content to download that's available on your console. But we're going to get to those questions now. So for the first, what are the key parameters and variables considered in silico modeling other than the net charge and chemical bonding? And can in silico modeling assist in the design and optimization of filtration as well?
Until now, significant result has been found in net charge and chemical bonding within the model. Moving forward, we will continue to explore additional parameters for the in silico model by leveraging historical data. Regarding the filtration process, given its focused on size or concentration, utilizing in silico analysis may not have a significant insight.
All right, thank you. Moving on, does Samsung Biologics have a resin library that can be utilized to determine and recommend the most optimal resin depending on the molecule type?
Certainly, Samsung Biologics has a resin library and advises on the most optimal resin for the client molecule. Once the resin is confirmed from both clients and our team, we can proceed with the subsequent steps. To elaborate more on this, in the case of monoclonal antibody, we recommend using Samsung Biologics platform resins, PA, CX, MMX. The performances of these resins have already been confirmed in previous projects. In case of bispecific and FC fusion, we recommend to use resin derived from resin screening. I think it is the best way to select the resin after understanding of their own characteristics.
Right. And what is some of the equipment that Samsung DSP possesses for resin screening or column process development?
We have Acta Avant and currently developing equipment with small columns to accelerate process development.
Okay, and let's see, I think we'll take one more here. Could you provide insights from the perspective of a downstream process expert on the tech transfer process for antibody products?
From the perspective of the downstream process expert, successful technology transfer for antibody product requires a comprehensive understanding of critical process parameters and effective risk assessment with mitigation strategies. To expedite timeline while minimize risk, utilizing platform processes is ideal. However, in the absence of platform process, early assessment of manufacturability and proactive management of raw materials are important. These proactive approaches will enable smooth transitions and enhance the efficiency of the transfer process.
All right. Well, thank you so much, Song-Young Kim, our expert from Samsung Biologics. If you would like to get a hold of him, his contact information is on the screen now. If we did not have a chance to address your question, we will be in contact with you. And to everyone who joined us today, thank you so much for your time, and we hope to see you again soon.